Every year, the ocean absorbs 25% of human CO2 emissions, which is a really huge service the ocean provides us, in that it's keeping that extra CO2 out of the atmosphere. If the ocean wasn't taking up that extra CO2, we would have already overshot a key threshold, and that's 1.5 degrees Celsius of atmospheric warming.
This is Adrienne Sutton, oceanographer at the United States National Oceanic and Atmospheric Administration. We brought her voice into this episode for a reason. Because today, we are going beneath the surface, into the ocean we don't see, where microorganisms work in silence, keeping our planet in balance. Despite the extra CO2 we keep depositing into the atmosphere, as a consequence of the economic model we live in. Every trip you take in your car releases CO2. Most of the products you buy were made in factories that release CO2. And every day, over 100,000 flights take off around the world, each one burning fuel and releasing CO2 into the atmosphere. These sounds you hear in the background are the soundscape of any city in the world. They represent just some of the sources of CO2 emissions produced by human activity. In this episode, we will talk about what happens in the ocean at a microscopic level, as surprising as it is complex. It is a balance that allows the ocean to absorb that extra CO2 and act as a buffer between us and a planet that keeps getting warmer.
I'm Dr. Christina Roggatz. I lead a Freigeist group here at the University of Bremen, and in the Concentrate Research Center, I have a scientific project that I'm PI of.
My name is Luz María Mejía, and I am currently working together with geologists to try to understand the climate in the past using fossils.
To explain the ocean we don't see, I invited Dr. Christina and Dr. Luz María in their company. I will take you to two places from everyday life that I hope will help you understand the hidden ocean science: a kitchen and an orchestra. I am Juan David Escorcia. I report on the environment, the climate emergency, and the ocean, uncovering what's changing beneath the surface and what it means for all of us. This science journalism project was developed as part of the Frontiers Fellowship, funded by the European Union. As you may have heard in previous episodes of this podcast series, between February and May 2026, I did a science journalism residency at MARUM at the University of Bremen. In this place, Tuesdays are the day to share a coffee. MARUM holds a coffee session where scientists sit together and talk. It was there that I met the people behind Concentrate Project. Over coffee, they told me about sugar molecules and the ocean. That word, sugar, made me think of a kitchen. I contacted Dr. Christina and asked for a proper interview. Imagine a kitchen as vast as the ocean. There is movement, there is life. In this kitchen, the chefs are algae. They work silently, taking carbon dioxide and with the energy of sunlight, turning it into sweets, small carbon candies.
So up to now, we do understand really well the role of individual aspects of the carbon cycle. We know the carbon dioxide is taken up by the algae. It is sort of recycled and used, and through photosynthesis they produce a whole range of substances, including a lot of these sugar molecules. They are exuded into the ocean, and then they are used by the bacteria, by microbes, by marine fungi, for example, as well.
The candies are ready, and like in any good kitchen, the guests don't take long to arrive. The bacteria, they come in and start eating. And Dr. Christina explains it in a way that's hard to forget.
Because it's a little bit like sugar bonbons, to some extent, for the bacteria.
So they eat, they're perfectly capable. In fact, and this is where the mystery begins, If you take those same bacteria into a laboratory and put the sweets in front of them, they eat everything completely, without a trace. But in the real ocean, they don't.
And they are in part degraded. And if we take these organisms into the lab and we test them, they are very well able to degrade all these glycans. But out in nature, they don't. And that is exactly the conundrum that we're trying to solve. Why are not all the glycans actually completely degraded?
Something in that kitchen we haven't been able to see yet is stopping the bacteria from doing what they know how to do. And the sweets that don't get eaten don't just sit on the table. They stick together, they become harder, more compact, and then they do something remarkable. They sink.
The best way to describe what happens is a bit like we all know when we do Christmas cookies. So we mix the dough, and that's pretty much what the algae are doing. They're taking all the ingredients and make the sugars. So we make the Christmas cookie dough, and we all know that Christmas cookie dough tastes the best when you taste it fresh. So there is already some of the dough gone by the time that you put the cookies into the oven. You make the cookies, and then by the end of Christmas, it's very unlikely that there is a lot of the cookies left, because it's the same with the sugars that the algae produce in the ocean. There is a lot of people who really, really like these little candies that the algae produce. So there is bacteria, there is fungi, there is all sorts of recycling going on. But what is interesting is that there is quite a few crumbs of these Christmas cookies that stay left over. And we really try to understand, what is the leftovers? Why are these leftovers? Is it because they don't taste as good as the other cookies? Or is it because they have certain ways in which they look, or they are perceived that they can't actually be eaten properly? So we really try to understand what makes those Christmas cookies, the ones that are not taken up and that are basically ending up in the bin at the end of Christmas, or in our case, in the deep ocean.
Think about what that means. That carbon was CO2 in our atmosphere. The algae captured it, the bacteria didn't recycle it, and now it's buried, locked at the bottom of the sea. The ocean is holding that carbon for us. That is what a major research consortium called Concentrate is trying to find out. 14 research groups, 5 institutions across Germany, all working on the same question. If the ocean keeps getting warmer, will the bacteria finally eat what they have been ignoring for thousands of years?
They could thrive on it, but there is factors, and that is the core of Concentrate, to find out what factors inhibit the degradation of these glycans. The aim of Concentrate is, within the second and third phase, to actually enable us to make predictions, to really be at the resolution, the molecular level resolution that allows us to not only understand the processes, but also predict how effective they will be in the future. So in my project, for example, we'll start understanding how are the enzymes degrading the different sugars in different conditions. And then we will be able to predict if we have ocean warming, if we have ocean acidification coming to play, because that's also something we have to take into consideration. How does that affect the whole turnover of carbon, the whole degradation of the glycans back to CO2, or the sequestration of these glycans? And is this in favor, or is it even a bigger problem that we're actually approaching? And by being able to make those predictions.
To put it simply, this kitchen, which is our way of understanding what happens in the ocean, has gotten warmer. The CO2 is being stored in those sweets, thanks to the bacteria not eating them. But now, someone knocks on the kitchen door. It is the Concentrate project, and they are here to find out 3 things. Why the bacteria haven't been eating the sweets, what happens to those sweets now that the kitchen is getting hotter, and whether this kitchen, which is the ocean, will keep storing carbon for us, or release it back into the atmosphere. But before we leave this kitchen, Dr. Christina wants you to see something.
Once you take a microscope and you look at the algae swimming about and the bacteria just cruising next to them, and they are all thriving and they are all taking the CO2, they're all producing the oxygen, they're all substantial part of our daily lives. That actually then makes people also even more aware of how complex this system is. And that's something that we try and communicate.
Now I want to take you somewhere else. From the world of glycans in the Concentrate project to the world of organisms in the ocean called coccolithophores. Remember, this episode is trying to draw in your mind the picture of an ocean we don't see. But before we get to these coccolithophores, let me tell you briefly how I found them. In my first days at MARUM, I noticed a poster on the wall. It listed the names of scientists who volunteered as support contacts for colleagues dealing with stress and anxiety. One name caught my eye, Luz María Mejía. We don't share a surname, but I had a feeling she was Colombian, like me. Days later, someone on the MARUM team mentioned her and her research. I reached out, and as Dr. Luz María Mejía explained what she does, I began to think of the ocean as a vast symphony. One that has been playing for millions of years. Here at MARUM, scientists are reading the scores of that symphony in different ways, trying to understand how the ocean's great orchestra sounded in the past. She reads the shells, specifically the chemical bonds inside the calcite of organisms called coccolithophores, microscopic algae that sink to the seafloor when they die.
Okay, so we use fossils from very, very, very small algae that are called coccolithophores. So this is like a model of a coccolithophore. And you can imagine these are single-celled organisms, and each of these plates are formed inside the cell, like a pregnant woman, that they are forming inside the cell and they just push it out, and it forms many, many different plates. So these organisms, they are algae, and therefore they need sunlight to do photosynthesis, which means that they only live in the surface of the ocean. And when they die, so let's say the surface of the ocean is here, and they live here and they are happy, and they die and they start to sink. And when they reach the ocean floor, let's say that we have like the oldest coccolithophores down and the newest coccolithophores up. So we go and sample, we take like a tube from the sediment, and everything that is down will be older fossils and everything that is up is newer fossils. And we study the chemistry of the shell that these organisms produce to try to understand what was the temperature in the past, using an indicator. I am going to say the word proxy a lot of times, but what it means is like an indirect indicator of a parameter, in this case temperature.
These small organisms called coccolithophores are like tiny instruments in a vast orchestra, each one playing its note while alive. But you have probably noticed how a single note slowly loses its intensity in the vast sound of an orchestra. Like the one you are hearing right now in the background. Well, the same thing happens to coccolithophores when they die. They are no longer at the peak of their life at the ocean surface. They die, and slowly, layer by layer, they are buried in the deep ocean.
So when they die, they sink relatively fast, which means that the other, like bacteria and things that are trying to degrade it, they are not going to degrade it. And so a part of the carbon that has been produced up in the upper ocean is going to sink down and stay in the deep ocean, which means they're also capturing CO2. When it's down there, it takes a lot of time, thousands of years, to go back up because of the circulation of the sea. So in this way, they are connected also to climate.
These organisms don't just play a role in the ocean's carbon cycle. helping to capture and store CO2. Dr. Luz María Mejía also studies them to uncover the climate of the past. But studying coccolithophores is not a simple task, and this is where the music of our orchestra gets more complex. She studies the temperature of these coccolithophores by counting the bonds inside their shells, and what she knows is this.
So the amount of bonds that are formed in this calcite, in each of these coccoliths, between these rare forms of oxygen and carbon is only dependent on temperature. This is the theory. If it's only dependent on temperature, and we measure the amount of bonds that there are here, it means that if it was cold, we will have much more bonds. If it was warm, we will have much less bonds. So we are measuring the degree of clumping.
More bonds, colder water, fewer bonds, warmer water. And nobody had tried this with coccolithophores before.
We have discovered very important things, or things that we think are very important with this, because this is the first time that someone has ever tried to measure clumped isotopes in coccolithophores. No one has ever tried before me. So in that sense, we are kind of advancing in the community of this proxy. So on that side, we are going forward.
And this is what makes her work so remarkable. By reading those bonds in a way no one had done before, Dr. Luz María is contributing to one of the great challenges in ocean science, knowing with precision how warm or how cold the ocean was millions of years ago.
So we have started, at least to make people try to understand better what they are analyzing. This is the idea. Because making assumptions, or like giving all this information to climate modelers for them to try to understand what's going to happen in the future, it is very important that we are not only sure whether it was warmer or colder, but also how much warm or how much cold it was in the past.
And that matters, because if we think about it, the whole point of spending years reading the climate of the past comes down to this.
So the idea is to go back in the past at times in which we think we are going to go in the future, and if we understand how the climate responded to whatever CO2 there was in the past, maybe we can better predict where we are going in the future. So how much and how fast we are going to warm up different areas, surface areas of the ocean in the future, if we compare to times in the past that we think were similar.
In this episode, we invited two researchers, and behind each of them, the same uncomfortable reality. The ocean is changing faster than we understand it. And the science needed to understand it demands something that the climate crisis makes very difficult to justify. Time, Dr. Christina explains.
We need to be mindful and we need to be careful to really be sure about the results that we generate, and not very briefly and quickly do experiments to reach a conclusion that actually doesn't warrant the message and is not supportive enough or is not replicated enough. So one of the key things doing science is actually to replicate things as well and to really be sure about the outcome.
But keep in mind that all this marine science research is trying to solve problems that affect every single one of us. To give us a sense of how big this is, Dr. Christina reminds us of a number you already know.
So every second breath we take is made by the algae. But what they also produce at the same time is these glycans. And we try and understand if this is maybe a way by which a lot of the CO2 can be sequestered biologically through drivers that we can understand, and we can then also aim to predict, so we have an understanding of what is going on first before we are even able to start manipulating something to maybe aid reducing the amount of CO2 in the atmosphere, or at least finding ways of buffering the system to some extent.
Science can produce predictions about the future, as Dr. Christina describes, but that is only half the challenge. The real question is whether that knowledge ever reaches the people who need it most, and that is something Dr. Luz María has reflected on deeply.
But is this really important? Who's going to read a Nature paper? I just published a Nature Communications paper, and it is cool. A lot of scientists are going to read it, but it's not going to have a real impact on the society because normal people, policymakers, journalists, they don't read these things. I mean, I changed to work with white powder in a lab instead of working with sea turtles and corals in the second most biodiverse country in the world because I wanted to have an impact, not because I want to have millions of papers published in journals that only my peers are gonna read.
I am Juan David Escorcia. I report on the environment, the climate emergency, and the ocean, uncovering what's changing beneath the surface and what it means for all of us. This science journalism project was developed as part of the Frontiers Fellowship, funded by the European Union.